And in the meantime, so I'm delighted to, but now I'm not 100% up to date with your CV, Giselle. It's changed a lot, but I'm delighted anyway to to welcome Giselle Miranda to our course again. She used to give this lecture and workshop, and then there was a break for 1 year, and now she is a group leader at KTH and SciLife and works with image analysis and is also part of BIIF, the Bioimage Informatics Facility, which is based in Uppsala and in Stockholm. And they do a fantastic job at helping people build their pipeline, and I fully recommend to anyone. And I think they're also they cater for the whole of Europe, right? Because this is Euro bioimaging, right? So then I recommend to anyone to get help from them to at least get started. Giselle, please, thank you. And if I forgot
Yeah.
something about in your titles and stuff, you tell me.
Yeah, I think it's nice to be back. I think I've been collaborating with LCI since 2020 on this course. Yeah, last year I couldn't be here, but it's nice to be back. And I know you've been on a long journey in this course, talking about imaging, different imaging modalities, sample preparation, covering sample preparation as well. And then we have the last module on bioimage analysis. So I hope you are still energetic. And I heard there is a pub for after this module
Uh,
as well.
I hope you can
So
come.
yeah, good. So what I bring here today is we are going to cover a bit what is bioimage analysis. We are going to see some examples. But to work with digital images, it's also good to understand basic principles, concepts on digital images. So we are going to cover a few topics as well. We are going to talk a lot about image quality, especially in the workshop, how to ensure image quality for image analysis. We are also going to cover some image processing, pre-processing techniques and enhancement. We are going to see some typical bioimage analysis workflows and an overview of machine and deep learning based approaches for image analysis. So uh, here I'm going to use bioimage or image analysis interchangeably. But of course, when we're talking about bioimagery, we want to understand and quantify microscopy. I included here microscopy images, but we can include as well medical image data. But typically we have here an example of fluorescent images from the Human Protein Atlas. So you are interested in tagging different proteins and you want to somehow, for instance, quantify a protein that is localized in the nuclei. And then the first step is to segment, separate the objects of interest. So you somehow obtain a kind of quantitative measurements to here we are tagging the intensity in the nuclei, so you are able to compare, right, across groups. That's a very common task. And so this process means that we are going from the image data, we're extracting some numbers, and finally deriving some knowledge of these images, right? And this is a quantitative process. So we want to arrive at some measurements. We want to make it quantitative. It's an objective process because we have a goal, right? So here I talked about proteins in the nuclei, quantifying proteins in the nuclei. And it's reliable because the method you are using to measure meets your goals, your objective. It's repeatable. So if you repeat the experiment, right, with the same images, you should be able to get the same results and reproducible. So if someone else is also doing a similar analysis from other samples of the same proteins should also reach the same results, right? So why we don't analyze manually anymore, I think I don't have to motivate it much here, but we have a high throughput data creation. So we have advanced microscopy techniques. We can think here about light sheet, different optical modalities, EM, screening microscopy. So we have also more fluorescent labels and sensors to tag different proteins, for instance. So we are in a scenario of high throughput data creation. And here you can think about volume data, of course, time lapse, but also nanometer resolution, which is impossible to extract any knowledge here just by visualizing or manually handling these images. But at the same time, we also have faster computing and better algorithms to deal with this large scale data. So, and of course, another reason is that if you have an automated framework, it's much less biased than humans. So, and much more reproducible as well. So we can say we are here at the intersection of computer science, because most of the time we need to either we need to use software tools, but many times we need to develop something as well or customize a solution. Also, a good knowledge of biology is required to interpret the data and being able also to derive biological hypothesis and interpretation about the data. And I would say, a good knowledge of statistics as well to validate your hypothesis. And so this field is also known as bioimage informatics. That was a term coined in 2010 and still very much used. So if we think about some examples, for instance, we can group some of these bioimage analysis tasks in cellular imaging. So in this example, for instance, we want to, the task here is intracellular localization of lysosomes in the context of cancer and normal images. So what we did here is, if you're interested in how they are spread inside the cell, we map the cell as a distance map. So you can see here each column as a different sample. And then first we create this distance map. And then from the specific channel that we are tagging, the lysosomes, we can now segment the lysosomes and then check how they are distributed within the cell. So we map here the distance. We have a histogram of the spatial position of the lysosomes in the cell. So here using, for instance, cellular imaging. But we can also think about live cell imaging. So here we are interested in dynamics. So this is an example of tracking chromosome dynamics in live yeast cells. It's a bit more challenging task, since we have also to track the same cell over time. But here, what we are doing here is we are tagging a specific protein attached to the microtubule during cell division. And here, I don't know if it's possible for you to see, but the shape is like a line. So we were able to track and then segment, and here is the reconstructed dynamics. And then you can extract, for instance, measurements like displacement over time or average distance, for instance. And the last example here is 3D image analysis. So of course, it's also interesting and to analyze samples in 3D. So here's an example of a single cell transcriptional roadmap of human embryo development. So here we are interested in analyzing the co-expression of specific genes in 3D. So here we have multi-channel fluorescence image, different tagged proteins. And so all these examples, we cover cells, but we can also think about tissue. And for tissue imaging, we are interested now, for instance, in analyzing specific features within the tissue, still being able to segment nuclei and cells. That's also a common task, but we may be interested in other cellular compartments and the specific markers as well. So when we are working with tissues, we are also trying to understand structure, function, and pathology of tissues at the cellular and molecular level. And you can also do things like neighborhood analysis to track spatial distribution of tissue components. And the examples are, for instance, here we have a structural analysis of cervical epithelial tissue. So it's very common. The epithelium has this distribution. It's distributed in layers. So many diseases affect different layers of this epithelium. So being able to characterize, for instance, the specific segment or separate the layers of interest, that's a very common task. But you can also think on multiplexed imaging. So new technologies that are now allows us to track a large number of proteins for the same sample. So you can have that's what we call a highly multiplexed imaging, for instance. And we also have stained tissue as well, very common used in the clinical practice. So here we have examples of tracking specific cells. This is an example of a disease that is called cerebral cavernous malformations. So we are tracking here, for instance, changes in cell neighborhood and other similar similar examples here as well. So here analyzing also epithelium, breast cancer, and here colorectal cancer. So analyzing different structures, neighborhood, and different features within the tissue. So I hope I have motivated with these examples. I will, in the interest of time as well, I will finish here with the examples. But you are welcome to ask questions about them or bring other examples also at the end of the lecture. Sorry, I saved one example in detail here. So just to illustrate the pipeline, so for instance, for the structural, this is the example of the epithelium, and we are interested in tracking how the layers of the epithelium change in the presence of HIV. So here you have more details, for instance, how first we select the regions of interest, so sections of the tissue, and the different channels that we have, and the steps required to extract the measurements here. So first, we need, of course, to separate the epithelium in relation to the other tissue here. Then again, we use like distance transform to separate first the three layers. So we have the upper layer, the intermediate layer, and the lower layer, and other intermediate steps. So each of these, or each of the channels here, will give us a specific part that is part of this the whole pipeline. And here also just to illustrate. So we go from epithelium. We use this transformation that is able to track these proteins that they form like a network. So what happens here is that there's a disruption of the tissue in the presence of HIV. So the height of this layer changes. So here we can see that we could segment this layer and then finally reconstruct here the model of the net. So when this net is disrupted, then we have, of course, the area occupied in the tissue. It's smaller, and then it allows the virus to go through all these layers. And here we also did some transcriptional profiles, so bulk RNA sequencing, and then what we found here is that there is we found differentially expressed the genes linked to biological pathways. So this from the bulk RNA analysis. So an upregulation of immunoregulatory genes and a downregulation of genes involved in epithelial barrier integrity. And these we also, which corroborated like the morphological analysis with our pipeline. And of course, from here, you can go to cellular, to individual single cell analysis, right? Also analyzing expression levels within each cell. This also just to, this is the last example, but just to give an overview, here's the example of multiplexed imaging. And here we wanted to track the expression of specific proteins associated with the transformation of adult spermatogonian stem cells into developed sperm cells. So this is testis tissue. And the idea of working, of course, with this multiplexed imaging is to create a kind of a spatial map of proteins. So you can see which proteins are, it's a kind of on and off map of which proteins are being expressed. And here in relation specifically like following up this transformation of sperm cells. One interesting thing here, we can see all the channels that we have. So it's the same image, but the different proteins. So we can see the variability. And one of the issues we have, like if we follow up with the histograms of each channel, is that it varies quite a lot, the distribution. So if we take this histogram, for instance, it's really hard to define what is positive or negative. So one thing that we did in this work is that manually we use the pixel-based classification approach, and then we first manually annotated what is signal, what is background, and what is a negative signal. And then we trained the model to obtain this map with each, like, to define what is the proteins of interest when it's being expressed. So this is also another example here. This is a semi-automated example. And then we can, of course, combine with cell masks. So we have the region that is being expressed, so the protein, and then combine with the labeled mask of the nuclei or cells, and then quantify intensity, for instance, the expression of these proteins. So yeah, now this is my last slide about the examples. So in all of these examples, pipelines, one thing that, how do we start thinking about this problem, right? Well, you have to start with your scientific question. So how does it involve image analysis? Do I need, what will I need? Is it segmentation? Do I need to get, do I need to analyze intensity for each cell or for each tissue component? And so this is really important because then you design the experiment according to your scientific goal. So in all these examples, we had a biological motivation, and then what we try to do is develop a workflow or pipeline to answer that question, right? And now we are going a bit on the basics of digital images. I know that some of the topics you covered during the course, but here is just an overview. So we need to remember that for all these examples, we are talking about digital images, and for the computer, all of these images are just a matrix of numbers, right? So if we take like a sample image here and we zoom in this area, what we have is actually a matrix of specific pixel values. So in Fiji, for instance, if you're familiar with Fiji, you can, there's this tool called Pixel Inspector. So if you click in a specific region of the image, you will get this, a matrix of values. So you have a certain number of rows, so certain number of columns, and a range of numbers here. But how are these pixel values defined, right? So from our sample, from the interaction of our sample with protons, Uh, the protons from our sample interact with the sensor, and we generating here some electrons, and this signal is amplified, and then we can convert then to a digital signal. So this is actually the analog digital converter. And you saw during the course, for instance, that you have different sensors, like detector-based sensors, camera-based sensors. So the pixel intensity here will also depend on the sensor type, the camera type. And we can think in an analogous way for color cameras as well, as here the biofilter now responsible for this conversion to the pixel intensities. Here we know that the green filter, for instance, is double the size of the red and blue filter, since the human eyes more can discern better the green image, the green line, the green signal. So so this is the process of obtaining the pixel intensities, right? Another concept that is very interesting, very important to understand when we are working with digital images, especially microscopy, is the bit depth. So the bit depth corresponds to the number of gray levels. So if you have only one bit to represent your data, you have either like a black or a white intensity. So you can represent only two levels. If you go to 2 bits, you can represent 4 levels, and this number is computed as 2 to the power n, as you can see here. But in microscopy, very common like 12 or 16 bit, and we can see the range, the number of grayscale values we can represent. The images, 8-bit images are very common as well. But microscopy usually we obtain we acquire usually images are 12 or 16. So the number of the number of gray levels here is the number of possible intensity values. So if you have 8 bits, you can represent values between 0 and 20 and 255, for instance. And more bits so represents more details and lower risk of saturation as well, among other artifacts. So you have a higher level of details when you have more, when you have an image with higher number of bits. But more bits are usually better, but you can also, there are some situations, for instance, when you have storage limit or dim samples that are lower bit is also desirable. So it's not that you cannot work with 8 bits, but this is something you should also think when you are acquiring the images. What are the limitations? Or do I need more bits? But usually, yeah, more bits, then you can represent a higher range of values, of intensity values. So here we are talking about grayscale. Before we talked about grayscale images. When we go to color images, like an H&E stained image, then we have RGB means here that we are representing this image in the red, green, and blue space. And we have one matrix for each of these colors. So now what we can see here is a combination within the RGB space. So this is an image acquired, it's a colored image. So different from the previous one, for instance. But we can, the same way, separate these channels and then analyze each channel individually. So we can see, for instance, what is highlighted in red, green, or blue. We can even process them separately. But another way, when you're working with color images, is instead of splitting the channels in the RGB space, is to use what we call a color deconvolution. So you take advantage of what we call optical density of the dyes you are using, in this case, a methoxylene using, and you try to separate the channels as a function of these dyes. So then you have, for instance, the first image here highlighting the hematoxylin stained parts of the image. The same here for the usine. And this can help, for instance, if you're segmenting the nuclei, this can be a better way to split the images. When we talk about fluorescent images, different from the color images, here we are talking about independent channels. And the color we have here is just a representation. Actually, they are gray level images, so you can treat them completely independent of each other. Still, we have the same matrix representation for each of them, but what this means exactly is that, for instance, you can represent this image, for instance, with different color coding schemes, what we call lookup tables. And why should we do that? Well, changing a lookup table, for instance, can help us define, identify regions of interest. So you can highlight components of the image, for instance. Examples of common lookup tables is, for instance, for labeling elements. So we use in Fiji, if you're familiar with Fiji, if you use this lookup table, you can, for instance, give a specific label to each component of the image. And this, of course, helps to identify individual components, especially if they are clustered together. It's not the case here, but when you have a clustered, like a nuclei, for instance, or when you are working with tissue images, this is very useful. Other examples include, for instance, detecting artifacts. So saturation and underexposure is an example. This is a lookup table called high-low. So you can see here in red what is saturated and in blue what is underexposed. And another important concept are histograms. As in statistics, the histogram is just showing us the distribution of the pixel intensities, right? So we have here different examples. Here's an 8-bit image where we have, when we can see here the minimum value, maximum, and the most frequent, for instance. So this one we can see it's a very bright image. So the peak here on the bright values of the histogram. And so on, the same for the other images. So the pixel order here does not affect the histogram, right? So the histogram is basically counting the number of pixels that having a specific pixel value. So it doesn't matter the order. And we can derive basic measurements, so like mean, variance, standard deviation. In the example of the multiplexed image that I gave earlier, we could see, for instance, how the signal is distributed. Can we identify a peak? When we have peaks, for instance, it's easier to separate background from the background from the signal of interest. And it's also important to understand the concepts of brightness and contrast. Brightness is related to the amount of light, and it's a relative context. So when we look at both images here at the same time, we can say that B is brighter than A. But contrast is related to the maximum difference in intensity. So, and here we need a histogram to visualize that. We can see, for instance, that here we have the maximum value is 166. So here's we are in the 8-bit world here, and it's pretty much dim. So when we enhance contrast, it's like we are rescaling the intensities now to the whole interval of the whole range available. And why do we do that? We can do that to inspect the image, or we can do that even as a pre-processing step to help with segmentation. And also, now more and more with high high content, high dimensional data. In microscopy, especially fluorescence microscopy, we talk about channels, volume, time. So it's important also to understand exactly what the dimensions of the image are. So for 2D images, right? So these are the examples we saw before, the matrix representation. But for 3D image or stack, then we have here a 3D matrix, right? But the next example, we have a 5D image. So this is a screenshot from Fiji, and here you can see how Fiji represents this what we call hyperstack. So we have here the channels, and also here you have some information here on the top on the upper part of the window. So here we have two channels. This is a mitosis example. We have a Z stack with 5 images, and we have a time lapse with 51 time points. So here we have what we call a 5D image, and that's the highest dimension we can have. Also, metadata is understanding metadata is very important. So usually you have from the raw data, you can access metadata information which corresponds, for instance, to things related to the experimental to the experiment, like protocols, probes, treatments, or the acquisition, the image acquisition, so parameters, instrument settings, and so on. But also other analytic information like outputs from processing and analysis. So in Fiji, for instance, you can access this metadata information. And this is also linked to file formats, right? So when we work with microscopy, there are different proprietary formats. So for instance, NED2 for Nikon or Zeiss format, but these usually can always convert these file formats to general formats like a TIFF, for instance. But here, the recommendation is, for instance, JPEG is lossy compression, right? So we lose information if you export to JPEG. So we never use JPEG for image analysis. TIFF is the recommended format to go if you need to export because it's a lossless format. It can also can also represent stacks in TIFF. And PNG we usually use to transfer or display data. So it's not possible we don't have stacks in PNGs as well. And other file formats for publications, for instance, that may be interesting, like for instance, a vector format, right? So here you can change the format without affecting, for instance, the final output here. So this is what we call a vector format. It's a vector representation of the data. And finally, just to highlight, because it may be a bit confusing to have all these formats, but there are specific plugins. This is in Fiji, it's called Bioformats, but that's a more general plugin that people working on other platforms can also have access to. And here it allows you, for instance, to open different file formats. So if you're working in Fiji, this is the interface you will see to import the image. So a good practice here is to always keep the original files and refer to the acquisition software for the original metadata. Well, once we have acquired the images and we guarantee the correct format, right? I want to move a bit here to ensuring image quality for image analysis. So some of what we call image artifacts they cannot be corrected by image analysis. So you arrived, for instance, at the last stage of your pipeline, you acquire the images, and now you want to analyze them. But these are some artifacts that cannot be corrected. So you need to try to mitigate them at the acquisition phase. So let's see examples of these artifacts. And I'm sure you already covered saturation, but here I just wanted to show the histogram corresponding to these three images. So we have the same images in three different situations. And we have here this very dark image. So you can see that the histogram is the range of values is here on the left side, so it's very dark. So here you lose, for instance, information on the edges of the objects. For instance, you cannot, for instance, like some parts of the bolt are mixed with the sea here. And on the right, we have the opposite situation. So here we have the overexposed situation. So the values are here more to the right side of this histogram. It's still hard to identify. For instance, we have a more homogeneous distribution of intensities here. So the clouds, for instance, are mixed with some parts of the bolt. So it's hard to distinguish here. So ideally, what we want is what we call a well-exposed scenario here. So we don't have saturation or overexposure. And this is something we cannot fix at the image analysis side. So we need to fix that during acquisition. Another thing is that these saturation can lead, even if you can still process the image, of course, but the measurements may be biased, especially in relation to size. Bleed-through cannot be corrected with image analysis as well. So bleed-through happens usually when you have like simultaneous acquisition. So we have the bleed-through situations, the same sample, but in the second row, we have the simultaneous acquisition, and in the first one, a sequential acquisition. So it's this crosstalk between fluorophores, and this can compromise intensity, shape measurements, and even like colocalization analysis. So when you want to analyze if the proteins are being expressed in the same region of the image. Also, bit conversions. So if you have an image that is 8-bit, here's an example of if you convert an 8-bit image to 16-bit image, some softwares, they allow you to do that. But so you are mapping a signal that much lower resolution here. There's a lower range of values to a higher range of values. So this will not it will not change the pixel values, right? But when decreasing, if you have a microscope image that is 16 bits and you want to decrease to 8-bit, then you can apply scale or not. But here you're also losing information. So in Fiji, you can control that in the edit menu with the conversions option here, with the conversions item. So the take-home message here is to be careful with these conversions. So take care when converting or changing the bit depth. Also, some image operations. So we talked about the contrast before. So if you enhance contrast, for instance, for better visualization or pre-processing, then always create a copy of the original image. Now Fiji, if you try to apply the contrast, Fiji gives you a warning that, hey, you are changing the contrast, the pixel values will be changed. So this can also be reversible. So always keep a copy of the raw data. But some artifacts can be corrected with image analysis. Sometimes it's better to correct them while acquiring the image. But for instance, denoising which is a very common issue. And inherent noise is inherent to some optical systems. So here we can see an example of denoising. So these are things that we can, the pre-processing steps in the image analysis workflow can help. Stabilization is another example. So here we have a time lapse. So we have an example of a drift in a time lapse. So this needs to be corrected as well before analyzing, before going further in the analysis of this image. So this can also be, there are different algorithms that can be used here, different registration tools that can be used to correct. And illumination is also another example. Here we have an example of a mosaic effect. It's a vignetting effect. And we can see the change, the differences in illumination in this image, and here the corrected version. So other examples here. So this can also be handled in the image analysis side. One example is the rolling ball algorithm for illumination correction. We are going to cover the algorithm during the workshop as well. But here we can see the differences in illumination from left to right. So if we change the lookup table here of this original image, we can see how the intensity is the gray levels here on the left are much darker. And this rolling ball, it's basically oh, and here it's also another representation here. It's just a 3D plot of the intensities for each pixel. So we have for each pixel in this image, the height represents the intensity. So we can see the trend here, right, from left to right. With the rolling ball, what it does is you can think about, as the name says, like a ball on this surface. And when you have a valley like this among these peaks, it will define the height on which, or the extension here, in which the ball can touch this surface. And then you can reconstruct these, of course, here it won't be able to reach this valley. So you can reconstruct only, this is the reconstruction of the parts of the image that the ball, of the surface that the ball can reach. But if you now choose a smaller ball, then you are able to reconstruct more of the surface. So you're bringing more signal to this background representation. I'm giving these two examples because one of the parameters of this method is choosing how big is this ball? What is the radius of this ball? And this is a parameter usually you need to set and try different values, because then once you selected that from the original image, we are going to subtract the reconstructed background in order to be able to correct the image. So we always, the recommendation here is to choose a value that is of radius value that is higher than the objects you want to keep in the image. Otherwise, you will also filter them out. But this illustrates here how we can correct issues like illumination, uneven illumination, for instance. Well, we talked about the basics of digital images and some tools like histogram and how to play with contrast for visualization or pre-processing, for instance. And we covered some image artifacts that can or cannot be corrected with image analysis. So now that we have this background, we can talk about going a bit deeper into the steps of pre-processing and enhancement. And it's very common that we talk about the filters, right? We can, the mean filter, median filter, even during acquisition, we can use some filters. So what are these filters? We can think about them like operations designed to modify properties of the image. So you have here your original signal, and then you apply a certain function, and then you have the filtered image, the output. And classical examples include denoising, edge detection, and Gaussian blur. And you may be wondering why I want to blur this image, why I want to apply this filter. And we usually do this when we want to obtain more homogeneous distribution of pixel values. This can help, for instance, also in separating objects of interest. So in order to be able to analyze that, we need to understand, we need to visualize the histogram of the image. That's why it's so important to understand how the intensity values are distributed. So, but maybe maybe one of the most used filters as a pre-processing step is convolution filters. And you can, again, you can understand then as we have here our original image, and we are going to apply a transformation. Here we are applying an operation that we call convolution, and that's where their name come from. And this operation involves a certain kernel. So here's an example. Here we have a part of our matrix of values of our digital image, right? And then we have a kernel, and a kernel is just another matrix designed with a specific purpose. This one is designed to sharpen the image. You can ignore what these values mean right now, but what we are doing here is that we are multiplying this matrix by each pixel. So if I take this red highlighted pixel here, I will look into its neighborhood. The size of the neighborhood is also defined by the size of the kernel. And then there are multiplications for each pair of pixels, series of values, and then we'll obtain a final value for this pixel. So for this specific pixel, we are going from 100 to 160. That's the new value. And what's the effect, right? These operations quite powerful. So here we have the example of the mean filter. So here's the kernel representing the mean filter. As I said, you don't need to remember this filter, but it's just to represent the convolution operation here. So and the mean filter, you're actually taking the average of this neighborhood. So here again, this value that was originally 23, it's now 12. So the effect is that you have your smoother version of the image at the end. But we do this for every pixel in the image. So again, we analyze the neighborhood again, and then we have the new value for now this pixel here, the 43. This is an example of the median filter, and it's similar, but the median, we are not calculating the average. We are sorting the pixel values, so from lowest to highest, and taking the median value, the value in the middle. So here we are not creating new values, but we are reorganizing the pixel intensities. And what is the effect? So let's compare both of them, when to use each of them. So here we have that situation, we call it salt and pepper noise, because we have these isolated bright pixels within a neighborhood that is quite dark. If you apply the mean filter, you are going to average out this region. So all the pixel values will be modified. But the median filter will filter out these pixel, because it will be it's our highest value, so it will not be it won't be the median value of this for this region. And here, for instance, for this specific filter, you would use for the median for this specific situation, you would use the median filter. So Median filter can reduce noise, no new values are created, but the drawback is that's more time consuming because you need to sort values for each neighborhood. So the filter, it depends on what you need. What is the artifact in the image, for instance. The neighborhood size can also influence the effect of the filter. So you can use neighborhoods where you only consider like this, what we call four connected. So you're ignoring the corners here, or you can use eight connected, or you can even have a bigger neighborhood size, a bigger kernel. So all of this affects the final result. So if we take this nuclei here from using this HeLa cells example in Fiji, if we increase the radius, the effect will be more pronounced. So this is also a compromise, that the size of the radius is also a compromise. Here's just an example. If you want thinking more about how these kernels are defined, this is one for edge detection, and we can see here that on the left, it's actually a subtraction from the right side, the left side from the right side. So we are detecting, here's a horizontal detector, and here's a vertical detector. So we are subtracting like the top row from the bottom row, and this can help identify the edges. Other famous operators are edge detectors as well. So it's also possible to customize these kernels in Fiji. And finally, want to bring the Gaussian one as well. It's very common. This is very much used. This is an example to blurry images, and as I mentioned, you can also and make the pixel distribution more smooth for a specific region. The kernel here is Gaussian, of course, but you can also control here with the standard deviation, the size of this kernel. It's rotationally symmetric. Some advantages here in terms of computational efficiency. But here we have an example, yeah, the effect of this filter. So you can control how much you're blurring the image with the standard deviation parameter. So if you're working with Fiji, that's the parameter you will have to define there. Sylvie, how much time I have? 10 minutes?
Hi, Giselle. Yes, let me see. Yeah, it was supposed to be from yeah, 10 minutes more. Yeah.
Okay. So yeah, I will just go through the last class of filters here, and then we are going to talk a bit more on the workflow. So morphological filters can also help us detect shape. So common filters are the common operators here are dilation and erosion. So you can increase, for instance, here we have two objects, and if we apply what we call a dilation by B, so this is also a type of kernel, then we can connect these two objects. And the erosion can even delete some objects here because the kernel is almost as the size of this other element here. So we can also, here is a class of filters, as I said, that we call morphological filters. So you can perform these operations like dilating objects or eroding objects. And this can be done both in the binary cases, when you have, for instance, binary, the results of segmentation, and grayscale images. And here are other examples. These two operators can also be combined to form other interesting effects or pre-processing steps in imaging. So removing dark structures or highlighting bright structures. For instance, gradient is a kind of edge detector as well. Top hat for enhanced bright or dark structures, for instance. We can also filter in the frequency domain with the Fourier transform. So all the examples we talked about, we are in the space domain, but the same signal can be represented as in the frequency domain. And then we can apply what we call low pass and high pass filter. But the idea here, this is the representation of the frequency domain of the same image. This is actually the representation in the frequency domain. Here is the filter itself. So a low pass will allow like the only low frequency signal to remain in the image. And the low pass is a kind of has the same effect of smoothing out the edges. And the high pass the other way around, it will highlight the edges because the transition regions here we have a neuron, right? So the transitions like between the neuron body here and the background, this is a high frequency signal. So it's also possible to do this in the frequency domain. But now, combining all of this in a workflow, we have here a few steps you covered in the rest of the course on sample preparation in image acquisition, and I'm really being I'm summarizing here, but these are yeah, there's a lot to talk about here as you discussed in the rest of the course. And then we have pre-processing, so where we can use like this filtering, background reduction, subtraction, noise reduction, filtering. So all these concepts that we saw before. And why we do that? Why we do all of that? Because sometimes segmentation is one of the main tasks in this pathway, because often you want to separate objects. You want to separate not only cells, but parts of the tissue. So separation of the signal of interest is what we call segmentation. And you can also think about a refinement of this segmentation as a post-processing step, like labeling, eliminating objects that are, in the border that you cannot see then completely in the image, in that field of view that you have, right? And finally, quantification. So, and one thing that I like to highlight as well, since we are more and more like using AI tools, is that they can be used in different steps of this pipeline. So one example here of this workflow would be quantifying a certain, here in our case, in this example, beta-catenin translocation from cell membrane to cell cytoplasm. If you want to do that, the first step would be to split the channels. So here we can see each signal individually, and we can obtain the boundary. So we need to segment. Since we want to analyze, quantify the signal in a specific regions, we first need to obtain these regions. And then label in individual components, and then extract the measurements, right? So here we are able to quantify and compare. Is it different, the intensity in each of these regions? So the workflow here the workflow goes from splitting the images, getting the outlines of the nuclei from the DAPI image, getting the outlines of cytoplasm from actin image, and then measuring. And what we call, so separating these objects of interest is we also have this, we call them regions of interest, or ROIs. That's a very common term as well. And you can do this manually. So Fiji has tools that are like the ROI manager that allows you to do that. Or they can be obtained via segmentation. So in the example of the epithelium tissue, we had to first separate the epithelium from the other tissue. So here, our region of interest is the epithelium. But it may be like the border of the cell, of the nuclei or cells, or even it's very common that you want to, first you are interested in a protein that on the cell membrane. So you can define this ring as a region of interest on the cell border. So segmenting means separating the objects of interest, but the outcome is usually like this binary mask, right? And then which we can label later on. Thresholding is one of the most common methods. In Fiji, you can try different segmentation methods. And it's not that for all the images, if you have a set of images with similar samples, we recommend choosing one of these methods and applying it to all the images that you have. It doesn't mean that you always have the same threshold value, but you are applying the same process to each image, and that's so you're not biased. It's a less biased way to choose this thresholding. But you can see that if you choose different methods, you also get different results. There's no right or wrong. There is the method that will fit better to the purpose, to your purpose, to what you want to quantify. OTSU is very common. It's a method that tries to maximize the interclass variance in relation to the intraclass. So here, for instance, we have a pic. It's a histogram. So for each pixel intensity, we calculate this interclass variance. And interclass here, what we're calling class, is the background and the foreground. So here, clearly, we have a peak on the background signal here, and this is what actually we want from the image. And not always we have this nice separation between foreground and background and background, and then this can be a bit trickier to analyze the images. I'm almost over time. I just need 5 minutes more. So pre-processing, you can see that we segmented objects, but we need here to fill the holes or eliminate objects, for instance, that are not completely here, that are missing parts, for instance. So that is also posed as a post-processing step. We'll skip the basic arithmetics, but you can combine different combinations of these segmented masks for obtaining regions, for instance, that are only like the cell cytoplasm or only nucleus. And so what I wanted to show here is that in this pipeline, we have here the nuclei pipeline, let's say, and the cell pipeline. And the cell pipeline was not that good. So you can still, we need to optimize this pipeline. But for the nuclei, it seems it worked well, but for the cells, we have missing parts. So can we do better? And here we can use, for instance, other methods or AI-based methods that perform better than thresholding. And then, of course, once we have masks of our objects of interest, we can calculate the measurements. But with this last module, I just wanted to give an overview of what you can do with more advanced methods in the case of when you have the situation of the cell that you couldn't obtain a very good segmentation. So these are methods that you can think about methods that learn from data. We have different concepts here of these tools. So not everything is deep learning, not everything is machine learning. But this is actually the correspondence here with just to group these methods. But in machine learning, similar to our pipeline, we usually have our inputs and we try to extract features. And the features can be the measurements that we just saw from our binary mask, right? And if you have a situation that you have two classes, for instance, cancer, non-cancer, you can continue here using, for instance, a certain classifier to map objects of interest, to classify objects of interest. But now but in the last 10 years, we had a huge advances here on deep learning techniques where you not necessarily have to extract features independently, but you can combine here feature extraction and classification. And examples here, so you can think, for instance, how these methods work. You can think here you have a large data set. They are usually data hungry. So what you have to provide is your original image and your annotated data set here. What is the nucleus, for instance? And then you will train a model that will when you have a new instance, for instance, when you have your test data that was not seen before by the model, then it will be able to perform segmentation if you have a segmentation task. But this is this requires GPU, so it's computation hungry. And you can do these for not only segmentation, you can think about other tasks like denoising, for instance. So now we have our pair, training pair here is denoising. It's the noise image and the denoised output. And then now you are able, like for a new test data, you're able to apply the model, obtain a denoised image. So now in our pipeline where we got stuck here, we could try other methods. And now this is the result, for instance, using a software called CellPose. So it performs better than the thresholding, for instance. And this case is challenging because the cells are clustered. Here are other examples. I will stop here, but just as a last information, like as I mentioned, this is the CellPose method. There are other methods out there. And here's examples of, for instance, here's a comparison for this DIC image between CellPose and thresholding. So we can see we can perform better here on this examples. Other examples like segment anything for microscopy, and maybe one that is interesting for you working at Fiji is pixel-based classification. Here the idea is that you manually annotate given a color, so different colors represents different regions. And here, this is a machine learning-based classifier, and then you can obtain the final segmentation. So for each color you defined, you are training at the pixel level, what should be the output. I'm really over time, so I will stop here. But if you have any questions, yeah, let me know. Sorry for
Good. Okay. Thank you,
going.
Giselle. Thank you, Giselle. So we're going to take a little break of 10 minutes. So anyone who wants to take a break is welcome. We will come back at 25 past. And if anyone has questions, you're also welcome to ask now. We will anyway discuss
Go
during
back.
the workshop as well. Yeah.
Okay.
Yeah.